discuss about an activity which comes under chapter digestion and absorption. So this is basically about Visking tubing model of absorption. You can say it's an activity or practical. So here first we'll take a whisking tube. Now this is almost similar to, if I relate it to digestion, it is similar to walls of alimentary canal. So now walls of alimentary canal, it has tiny holes in it. So tiny holes means that only small molecules can pass through it, but not the larger ones. Big molecules can't pass through it. So first whisking tubing is somewhat like this. So first we'll take a whisking tube, then we will moisten it with water. Let's say we take a hot water bath, water is here, then we will heat it up. This is triangle, this is a sign for heat, with the help of Bunsen burner, and then we'll let just little bit heat, then we'll put that whisking tube here and it will moisten. So we will tie a knot in the one end and then we'll use a dropper pipette. Dropper pipette is like this. So first we'll fill this whisking tube with the help of dropper pipette, and here we will pour starch solution and then sugar solution. So after that we'll tie it and then we'll rinse that tubing in water here, like it's shown here. So then we'll rinse it with water so that any leftover starch or sugar is there, so that can be rinsed out. So now we'll put the tubing in a beaker. So we'll add enough water to the beaker so that tubing should be covered, then we'll leave it about for 15 to 20 minutes. Now we'll take a sample of starch and sugar from water. Now what happens, there is a process called diffusion. So there's a slight difference between diffusion and osmosis. Diffusion occurs in liquid, gas and even solids. Osmosis is limited to liquid medium. So osmosis, if I talk about osmosis, it requires a semi-permeable membrane, whereas diffusion, it does not always require a semi-permeable membrane, it's simple diffusion. Osmosis, it depends on number of solute particles dissolved in solvent. Solute means, let's say if I have poured something in it, this is water, this will be solute and this liquid part is solvent. So it depends on number of solute particles dissolved in solvent, whereas in diffusion it depends on presence of other particles. In osmosis only solvent molecules diffuse, whereas in diffusion both solute and solvent diffuse. So osmosis, it flows in one direction, whereas diffusion, it flows in all directions. So now this whisking tubing is like dialysis tubing. So basically, now this here we'll talk about whisking tubing. This is basically artificially semi-permeable membrane. So basically it's used for separation technique. So now what we'll see that here process of diffusion will take place. So it's like let's say this is a cell membrane and it is selectively permeable. So here automatically some gaps are there. These are gaps here. So see the size of this gap. So that means large molecules can't pass through it, only small molecules can pass. So diffusion here is passive, that means no energy is required. So now what happens? Let's say this is a viscous tubing. So here we have water plus iodine here we have starch molecules. So if I just draw this diagram in terms of diffusion, so it will be like this. Here we have starch molecules, and here iodine. So we know that starch molecules are large. So now these small molecules will move towards this. So the color will change to blue. So that means presence of starch is there. So now how this takes place? Starch plus amylase plus iodine, this will give us a blue colored solution. Blue color means the color will change, that means starch is present. This is basically test for starch, for presence of starch. Similarly, we have test for sugar also. starch, and for sugar we will add Benedict reagent. So the result will be orange red precipitate. That means glucose is present. If there is no formation of precipitate, that means sugar is absent. So these are basically tests for starch and sugar molecules. Now coming to questions. So first question is, why it is important to tie both ends of tubing. So see, string is of 1 centimeter. So now it is important because, or you can write, so that nothing leaks from the tube. Now coming to another question, why it is important to wash the outside of tubing. Now it is important, or it is necessary to wash outside of tubing to remove impurities or contaminants that may be present on the surface. Since impurities affect results of experiments, so viscous tubing must be washed before it is placed into boiling tubes, and also because it prevents cross contamination. Cross contamination means like between different solutions or samples. If the tubing is not washed, there may be leftover residue or particles from a previous experiment that could mix with new solution. So and they will give inaccurate results. Then it is important to wash outside of tubing to ensure accurate measurements. So any residue or impurities, they can affect flow of liquids. So when they will affect flow of liquids, that means variations in experiment can be there. So any residue or impurities and they affect flow of they affect flow of liquid, so it results in variations in experiments. Okay, now coming to another question. Which nutrient, starch or sugar would be which nutrient, starch or sugar would be able to get out of tubing? So they are asking which will come out, starch or sugar. So answer is sugar. See, starch molecules are too large. Why not starch? Because starch molecules are too large to pass through the tubing pores. So So this is because tubing is made of selectively permeable membrane. So glucose, or you can say sugar, these molecules are small in size, so they will pass because small molecules can move from inside the tubing to outside. Okay, and now reason is diffusion. And then coming to next question. Question number 4. Use what you know about starch molecules and sugar molecules. Okay, then question number 5. Imagine you have eaten a meal containing starch and sugar. Will both be digested? Both will not be digested. What will happen? Starch molecule is like this, a big molecule. Complex molecule, it will get broken down into small sugar molecules. So this way absorption takes place. So all the questions of it are done. Okay dear, thank you.